Every day, passengers arrive at Zurich Airport. In addition to the many people, there are also dangerous plant pests that have hitched a lift on imported plant products. Such consignments are inspected by the Swiss Federal Plant Protection Agency, whose job is to prevent particularly dangerous plant pests from entering Switzerland. I'll show you an example. On this mango, I've discovered a suspicious-looking fruit fly larva. If this turns out to be a particularly dangerous plant pest, I'll have to reject the goods. I can't identify the insect precisely through a visual inspection alone. That's because there are related fruit flies that are similar looking, but which are not regulated. In order to be able to identify the insect, it must be examined genetically. Until now, we would send suspicious samples to Agroscope's diagnostic lab. Researchers analyzed these samples using molecular genetic methods. This analysis was complicated, and as the samples were sent by post, it took us 2 to 3 days to get the results. This meant a long period of waiting for the fresh plant products, which could lead to a deterioration in quality. Now we can analyze suspicious plant pests right here at the airport. We use a quick genetic testing method to do this. Now the findings are available within an hour. This means just a short waiting time for the products. The products don't spoil, and the import process is sped up considerably. The quick test, which is easy to use, works as follows. First, I transfer the suspicious insect material into an extraction tube. I boil it in an alkaline solution for 5 minutes at 95 degrees Celsius. I then mix the tube on the vortex. Since this can cause a small amount of the liquid to stick to the lid, it is then briefly centrifuged. Now I pipette a small amount of the violet extraction product into the appropriate reaction tube. In the following step, the tubes are mixed again and briefly centrifuged once more. There. Everything is ready. Now I place the reaction tubes in the analyzer and start up lamp at a constant temperature of 65 degrees Celsius. In technical language, this method is termed loop-mediated isothermal DNA amplification. It makes use of that fact that different species of living creatures differ in terms of their genetic makeup, in other words, their DNA sequence. This allows us to distinguish particularly dangerous, regulated plant pests from harmless, non-regulated organisms. Only when the lamp test detects the specific DNA sequence of a regulated plant pest does DNA replication take place. The analyzer can measure this replication, thus making it clear that the sample contains a quarantine organism. If, however, no DNA replication occurs, we know that the sample contains a non-regulated fruit fly species. Within just an hour, it's clear whether the consignment can be released for sale. In case A, I can approve the consignment since I detected no DNA replication in the tube. In case B, I have to reject the consignment since I detected DNA replication. The suspicious sample therefore contains the oriental fruit fly Bactrocera dorsalis, which is one of the regulated quarantine organisms. Since it can cause major damage, its entry must be prevented. Thanks to this quick test, we all save time. Inspectors and importers due to the expedited import inspection, and consumers since they can further process and enjoy the products without delay.